This lecture is on water. In this lecture, we will discuss the role of aquaporins in water movement, describe and calculate net filtration within a capillary bed, and apply the concept of net filtration to the most common mechanisms of edema. Describe the mechanism of thirst, and in detail diagram and apply the mechanism of ADH with respect to water balance in the body. The main consideration for regulation of water are the availability of aquaporins, filtration or Starling forces, hormonal regulation. Hormonal regulation includes the drive for water intake, which is thirst, the regulation through ADH, and also alterations of sodium, particularly through the renin-angiotensin-aldosterone system, which we will discuss in the sodium lecture. Aquaporins are selective water channels that allow free movement of water across the cell membrane. Water moves freely between the ECF and the ICF due to aquaporin channels in the cell membrane. The ECF and the ICF, therefore, are at equilibrium. Water can also move paracellularly or between cells if the cell-to-cell junctions in that tissue allow, meaning that if there's tight junctions within that tissue, water may not be able to move between the cells. And the gradients also have to allow that distribution of water between the compartments. There is exchange between the plasma and the interstitial fluid as well within the extracellular compartment. Fluid is carried by the arteries to the tissues and removed from the tissues by the veins and the lymphatics. Fluid enters the capillary beds where fluid exchange can occur between the plasma and the interstitial fluid. Blood and lymphatic vessels then will remove fluid from the capillary beds and return it to the heart. All of this functions if the forces and the gradients for filtration are moving in the right direction. So we need to define two terms here. Filtration, which is fluid leaving the blood to the interstitium, and reabsorption, which is fluid re-entering the blood from the interstitium. So for our purposes at the capillary bed, we're going to discuss the movement of fluid with respect to the capillary, meaning that when fluid leaves the capillary, that's filtration, and when fluid re-enters the capillary, that's reabsorption. Of course, in other systems, we will define filtration and reabsorption with respect to other compartments. Whether fluid is filtered or reabsorbed between the plasma and the interstitial fluid depends on two types of pressures in each extracellular fluid compartment. Hydrostatic pressure is pressure due to fluid. We think of this as a pushing fluid, meaning it pushes away from the source of the hydrostatic pressure. With respect to the capillaries, this will be arterial and venous blood pressure. With respect to the interstitial fluid, this will be the fluid that's present in the interstitial fluid. Osmotic pressure is the pressure of solutes that draw fluid towards them. Particularly when we're talking about capillaries, we're going to look at albumin or the oncotic pressure due to albumin. I've created a little model here. Let's look a little more closely at this, and hopefully you'll be able to draw your own when we're done discussing this. So here what you see is that we're looking at the capillary with respect to the arterial side and the venous side. On the arterial side, we need to consider arterial blood pressure and the presence of albumin. So here we have outward pressures. Arterial blood pressure is a force pushing outwards. Oncotic pressure is also a force pushing outwards if there are plasma proteins within the interstitial fluid, because remember those solutes are going to draw fluid towards it. So here we have outward pressures on the arterial side. Inward pressures on the arterial side are oncotic pressure within the blood, drawing fluid towards the proteins in the blood, or hydrostatic pressure within the interstitial fluid. This we would consider with respect to the capillaries an inward force. We have those same forces on the venous side, where we have venous blood pressure and interstitial oncotic pressure being our outward forces. blood oncotic pressure and interstitial hydrostatic or fluid pressure being our inward forces. I know this is a little bit complicated. We're going to spend a little more time on this. Overall, you can see that we're looking at these pressures to determine whether fluid enters or leaves the capillary. So this is a process of net filtration. If you don't already have a piece of paper with you, you should grab one because you're going to want to draw this. So to determine whether fluid enters or leaves the blood, we're going to look at the forces of net filtration. In other words, what is the net amount of fluid that leaves the capillary at any given point in time? How much fluid leaves the capillary is net filtration defined as the difference between the outward forces and the inward forces on that fluid. Outward forces are when fluid leaves the capillary. An outward force would be the capillary hydrostatic pressure pushing fluid out, or if there are proteins within the interstitium that haven't been taken up by the lymphatics, then that will be an interstitial oncotic pressure that will also draw fluid out of the blood and into the interstitium. We're going to use HP for hydrostatic pressure and OP for oncotic pressure. We'll use a little C for the capillary and a little I for the interstitium. Inward pressures are fluid entering the capillary. Inward pressures would be the oncotic pressure, or the albumins within the capillary drawing fluid into the capillary. And another inward pressure would be any fluid that's within the interstitium, for example, that hasn't been picked up by the lymphatics. That interstitial hydrostatic pressure can also push fluid into the capillary, and that would be an inward force or a reabsorption force. If we put those together, we're going to look at the difference between outward and inward forces at the arterial and the venous side to see whether we have a net filtration or movement of fluid out of the blood or a net reabsorption or movement of fluid back into the blood. Before we get too stuck on all of these calculations, I want you to pause for a minute and think. At the arterial end, what should we have? And at the venous end, what should we have? So before you ever do any calculation, you always want to stop and think, well, if the body systems are functioning as they should be, what direction should I find when I'm done calculating this? Then you can kind of check your answers to see if they make sense. So what direction should the fluid move at the arterial end? If you said outward, you were correct. The fluid at the arterial end of the capillary should be bathing the tissues and bringing in the fluid and the nutrients and the oxygen to the tissues. So at the arterial end, we want filtration. We want a net outward force when you calculate all of these together. What about at the venous end? What do we want at the venous end? If you said reabsorption, you're right. You don't want fluid pooling in the tissues. That can cause edema or swelling, right? So on the venous end, we want the fluid then to be returned back into the veins so that fluid can return to the heart and recirculate. I'm going to zoom in here on the equation so you can write it down. Okay, so our net filtration is outward minus inward forces on the capillary. We're going to take our outward forces, which are the hydrostatic pressure of the capillary and the interstitial oncotic pressure, and subtract our inward forces, which are the oncotic pressure of the capillary and our hydrostatic pressure of the interstitium. When we do that, what we want is that at the arterial end, this is a nice diagram from your textbook. At the arterial end, we want to see filtration. We want to see that we have a net outward force. At the venous end, we want to see reabsorption. We want to see that we have a net inward force. So we should be able to calculate that given some values. So let's try it out. Here's a net filtration calculation from basic numbers given in your textbook. We have values at the arterial end and values at the venous end. Pause for a moment here. Create your own diagram that demonstrates filtration and reabsorption between the arterial and venous portions of the capillary bed, similar to that textbook diagram, and then fill in your numbers for outward and inward pressure. As you fill in those numbers for outward and inward pressure, diagram them within your drawing. Where is that outward and inward pressure coming from? What you should do then is take your outward, subtract your inward, and see what you get. Pause for a moment to do this, and then we'll talk about the answer. Okay, did you do it? So what did you get? Let's look at the arterial side and then the venous side first. Remember, overall, we want the arterial side to be a net filtration. So do we have a positive sign on the arterial side? Yes, we do. It's positive. That means we have a net positive force. The number you should have gotten when you took the outward and subtracted the inward forces here is about 9 millimeters of mercury. That's 9 millimeters of mercury pressure moving fluid into the interstitium from the arterial end of the capillary. Did you get it? Good. On the venous side, we should see a negative pressure because we want reabsorption. Did you get a negative pressure on your venous side? Yes. Here we have a negative, and it's a -8. That means we have -8 millimeters of mercury pushing fluid back into the veins to return to the circulatory system. Anybody notice something interesting here? If we have a positive 9 on the arterial side and a -8 on the venous side, what's going on? When we look at the difference between these two, there's about 1 millimeter of mercury left over between the arterial and venous side. Is that 1 millimeter of mercury going to hang out in the interstitium? Do you think that's a good thing? No. That 1 millimeter of mercury is going to be picked up by the lymphatics. So here what we see is there's a small amount of fluid left over that needs to be picked up by the lymphatics. Because what happens when fluid builds up in the interstitium? Edema, swelling. Okay? A couple of quiz questions for you. Let's apply this. Venous obstruction. Venous obstruction causes blood pressure to build up behind the level of the obstruction. For example, you have a venous blood clot, some kind of hepatic obstruction, tight clothing at the extremities, prolonged standing. What will happen to the net filtration if venous obstruction occurs? Pause for a minute and do this in your diagram. Block off the venous pathway. What would happen at the venous end? All right, did you get it? Here's the answer. The answer is A. An increase in venous hydrostatic pressure will prevent venous reabsorption and cause fluid to build up in the interstitium, meaning that you would have a lack of return of fluid to the veins. Then what do you get? Edema. So we have an imbalance in the Starling or the filtration forces. The answer to what do you get is edema. Okay? Next question. What about inflammation? Inflammation causes increased capillary permeability and loss of proteins from the plasma or from the vascular space into the interstitium. Some causes of this could be trauma, burns, tumors, allergic reactions, infection, any number of things that can cause inflammation. What will happen to the net filtration if capillary permeability increases? Pause for a minute and use your diagram to work this out. All right, did you get it? The best answer is A, although it's possible to argue for some of the other answers. A indicates that a loss of proteins from the blood will decrease the capillary oncotic pressure and reduce the reabsorption of fluid to the capillary. It will also increase oncotic pressure in the interstitium and increase filtration to the interstitium. So if you said D, you are also correct. So A or D, totally fine answers here. Either way, what do you get with inflammation? Edema, swelling. We're going to talk more about edema in the future, but here let's just define it. Edema is tissue swelling due to fluid buildup within the interstitial spaces. A localized edema is a limited site of swelling because of local injury, similar to the inflammation example we just gave you. A generalized edema is swelling throughout the body. A dependent edema is where fluid accumulates in gravity-dependent areas for standing or supine positions for long periods of time. Swelling and fluid accumulation is a problem. Not only is it painful because it presses on the nerves, it can limit nutrient, oxygen, and water diffusion. It can limit blood flow and cause ischemia. Patients will report swelling, puffiness, tight clothes and shoes, limitations of movement, and weight gain with edema. It's one of the most important physical signs that you will see. In your textbook, there's a very nice diagram that shows the mechanisms of edema. The most common mechanisms of edema are increased capillary hydrostatic pressure, decreased plasma oncotic pressure, increased capillary membrane permeability, and lymphatic obstruction. Let's look at this diagram more closely. Here you can see many roads lead to edema. We can have direct fluid movement into the tissues because of increased capillary hydrostatic pressure. That's this one. We can also have lymphatic obstruction that causes fluid movement into the tissues. Lymphatic obstruction can also lead to decreased transport of capillary proteins. That can cause increased tissue oncotic pressure. That will draw fluid into the tissues and also cause edema. An increase in capillary permeability, as we just said, for inflammation, can lead to a loss of albumins to the interstitial space. More albumins in the interstitial space, edema. Decreased synthesis of plasma proteins, usually due to some liver dysfunction. Increased plasma fluid retention can also dilute the plasma proteins. In that case, just like losing plasma proteins, dilution of plasma proteins can cause a decrease in capillary oncotic pressure. If you decrease that capillary oncotic pressure, you have less reabsorption, edema. So many roads lead to edema. These are very, very important mechanisms to understand because when you see a patient who has edema, you're going to have to start to become that investigator that thinks, okay, what do we have here? Do we have a problem with albumin? Do we have a problem with inflammation, a problem with lymphatics, or a problem with our capillaries? You're going to have to sleuth through those to figure out the cause of that edema. Thirst is also very important for regulation of water. Thirst is the desire to seek out water or fluids, which is a key component of restoring lost volume and fluid homeostasis. Some conditions like diabetes have excessive thirst associated with them. Excessive thirst is polydipsia. There are also certain brain pathologies, psychogenic mechanisms for unclear reasons that cause an increase in thirst. What triggers thirst? I know thirst sounds so simple, right? It's an everyday thing. But actually, physiologically, it's quite complex. So thirst is triggered by serum osmolarity, sensed by both peripheral and central osmoreceptors. Low blood pressure can also trigger thirst. This is sensed by peripheral baroreceptors. Hormone and medication effects can also trigger thirst. One of the most important and strongest hormones for triggering the thirst mechanism is angiotensin II. We're going to talk about that when we talk about sodium regulation, because the RAS pathway activates angiotensin II and is part of the sodium balance system. What about water balance through hormones? The hormonal regulation of water is through ADH. ADH is also known as arginine vasopressin, or AVP. ADH is produced by the brain, made in the hypothalamus, and released by the posterior pituitary, an extension of the hypothalamus. Whenever you think about hormone regulation, you want to write out the pathway, just as we did for our signaling mechanisms and our homeostatic mechanisms. Think about the stimulus, the detection, and the response for that hormonal pathway. For ADH, the stimulus is increased osmolarity. The detection are osmoreceptors within the hypothalamus. When those osmoreceptors detect an increase in osmolarity, or too much solute relative to water, they're going to cause the release of ADH. ADH will then respond by traveling to the kidney through the bloodstream and causing an increase in water reabsorption or return of water to the blood, blocking the release of water in the urine. How does it do that? It increases aquaporins in a tiny region of the kidney that's responsible for the final portion of fluid balance in urine production. We'll talk more about these regions of the kidney, the distal renal tubules and the collecting duct, when we do the renal unit. What I want you to hold on to right now is that when ADH is produced from the hypothalamus, it goes to the kidney and blocks urine water loss. As it does that, it increases water reabsorption back to the blood, returning water back to the blood. You will also hear that ADH causes vasoconstriction, and that's part of its second known name, which is arginine vasopressin, or pressing on the vessels. It has to be very, very high levels of ADH to get vasoconstriction. It acts through a different receptor than that receptor that's in the kidneys, and that receptor is much less sensitive to ADH, meaning that you need very, very high levels of ADH to get vasoconstriction. When I say very high levels, I'm talking about the kind of difference in osmolarity that would be happening during, say, severe shock conditions. Not this day-to-day adjustment that you see normally with ADH in the kidney. So ADH is always acting in the kidney for these small adjustments in water reabsorption, but at very, very high levels in extreme conditions like shock, ADH can also cause vasoconstriction and help to restore blood pressure. In the next lecture, we're going to talk about sodium balance. Remember that sodium and water balance are very tightly linked. It's very common for students to mix up ADH and the effects on volume balance in the RAS pathway. So the little trick that I say is ADH2O. ADH2O, ADH is responsible for water balance. All right, that's it for this lecture. Let me know if you have any questions.